from my PhD research at the University of Ottawa with Reese Casson about diversification in Pseudomonas aeruginosa. And as you may or may not know, Pseudomonas aeruginosa is a crazy microbe. It is one of the most abundant organisms on the planet, in part because it can inhabit a very wide range of environments. It respires aerobically mostly, but can also do so anaerobically. It can catabolize a wide range of organic molecules, and so it's very good at adapting to many different environments. It's also an opportunistic pathogen, so it infects people who are immunocompromised. It's a very common problem in hospitals, causes a lot of infections there, individuals with AIDS. It infects burn wounds, and the types of infections you saw in the previous talk. But most importantly, it infects the lungs of cystic fibrosis patients. And there's lots of other things going on in the lung. There's lots of other microbes present, but Pseudomonas aeruginosa becomes the most common microbe around adulthood in all cystic fibrosis patients and remains there and for the rest of the patient's life. And it's very difficult to eradicate these infections. And there's many common adaptations of this bacteria to the lung environment. I've listed a few here: loss of virulence to avoid immune system detection, antibiotic resistance, a loss of motility. And something that interested me in this research that looked at sort of temporal samples of this bacteria from human lungs, they would often look at the single most common morphotype and look for correlations between that and symptoms, exacerbations of symptoms. And there was this distinct lack of correlation between a specific phenotype and exacerbation of symptoms. And this, a couple years ago, this led people to think that the diversity of this microbe is an important part of the picture. And so this shows, if you just look at the strains with the red triangle beside them, so these are the same clone from a single patient sampled over many, many years, and you can see that there's a lot of evidence that this diversity is arising in vivo in humans, and that it might play a very, very important role in adaptation of this microbe. And so I wanted to know, okay, what's driving this diversification? And the lung is a very ecologically complex environment, and so one hypothesis is that the actual physical structure of the lung is driving this diversity. There's two different lungs, there's an upper respiratory tract, so there's different places this can happen. It's also an ecologically complex environment nutritionally, and we know that Pseudomonas aeruginosa can use a wide range of resources, and so the abundance of different nutrients available in the lung could play a role in this diversification as well. And so I wanted to know what is driving the diversification of these populations in the lab. And so I wanted to test the effects of these two variables, spatial structure and nutrition, on diversification. And my research question is, what are the relative effects of spatial structure and nutritional complexity on diversification? And to do this, I designed an experiment with these two variables and two treatments of each variable for a 2x2 factorial design. I manipulated nutritional complexity using this medium called SCFM, which is synthetic cystic fibrosis medium. And it's a medium that we can use in the lab that has the same concentrations of chemicals, salts, amino acids, and macromolecules, all the things that are present in the lung in the same concentration. And we know that it's not a lung, of course. There are many things missing, like an immune system, for example. But it's the closest thing that we can get to mimicking the lung environment nutritionally in the lab. And then I contrasted that with just a simple minimal media. To manipulate the spatial structure of the environment, I simply added mucin, which is the protein in mucus that makes the medium more viscous, just makes a sort of thicker environment. And so within these 4 treatments, I evolved 30 populations for each treatment for about 220 generations. Again, again, to ask what are the relative effects of spatial structure and nutritional complexity on diversification. And you know, I expected to see the most diversity arise in the populations with both the complex nutrition and the spatial structure, and the least diversity to arise in the simple minimal media without the spatial structure, and an intermediate amount of diversity unknown in the other two treatments. And so I measured diversity in two different ways. The first way is I looked at all the populations, and I looked at the colony morphotypes that were present in all those populations after 220 generations of adaptation. And then the second way to measure diversity is I took individual isolates from 12 populations of every treatment and measured several phenotypic traits of those isolates. So I'll talk about the first one first, as you do. And so I scored the types that were present based on these criteria here: pigmentation, opacity, iridescence, etc., and asked in these populations, okay, after adaptation, how many types are present? And this is what I found. In a large majority of my populations, there were about 2 types present. A handful had 1, a handful had more. And so this is all 30 populations within each treatment. And I did see that the highest mean number of types was in the SCFM plus mucin treatment. And statistically, I found that the nutritional complexity axis did affect the mean number of types present. And so the SCFM treatment did increase the number of types, but that the spatial structure axis was not significant. And overall, you know, this wasn't the result that I expected. I expected to see much larger number of types present in the SCFM plus mucin treatment, and so I didn't really see this. But what I did see when I looked at the populations was that in the 2 treatments without the mucin, so the 2 treatments without spatial structure, I noticed that very similar polymorphisms were present within those treatments. And so in the SCFM without mucin treatment, I would commonly see an ancestral-looking type with a small colony variant. And in a large majority of the populations, this is what I saw. And in the minimal treatment without mucin, same thing, but a different polymorphism. So I would see in a large majority of the populations that there was a clear polymorphism between colonies that had autolysis and colonies that didn't. And on the other hand, in the 2 treatments with the mucin, I saw all sorts of different polymorphisms present in different populations. And so some would have a small colony variant polymorphism, some autolysis, some other polymorphisms present, but there was no clear pattern. And so I turned to the, you know, less subjective data than looking at morphotypes. And I took, as I said, I took 12 populations of each treatment, 12 isolates from each of those 12 populations. So 576 isolates, and I measured these 7 phenotypic traits of all those isolates. So I measured growth rate, pyocyanin and pyoviridine, which are toxins that this bacteria makes, production, antibiotic resistance in ciprofloxacin, 2 types of motility, spoon motility and twitch motility, and biofilm formation. And I'm only going to talk about 3 of these because of time. And so this is what I found. So this shows so each point here is an isolate, and this is pyocyanin production for each treatment, and each color is a population within the treatment. And so there's, you know, 12 dots of each color in each treatment and 12 populations. And so, you know, I saw that in the SCFM plus mucin treatment, an increase in pyocyanin production. Interestingly, for biofilm formation, I found a very dramatic effect in the SCFM plus mucin treatment, which is the most, you know, lung-like environment here. So this was kind of, you know, this result conformed to expectation, which is always nice, but, you know, twitch motility didn't. And so here I would expect the 2 treatments with the mucin to have the lowest motility because of the spatial structure. You know, we would expect loss of motility to be more common there, but I didn't see that. Instead, I saw the 2 SCFM treatments with the lower motility. But as I said, I cared about diversity, not just adaptation. And so instead, I wanted to look at the variance within the populations of these phenotypic traits. And just to remind you, what I expected in terms of diversity is that the highest to be in the SCFM plus mucin, the lowest in the minimal treatment. And what I found, so these points are, so each point is the variance of that particular phenotypic trait within a population. And this is for pyocyanin production. And here, I do see the highest amount of, on average, variance for the SCFM plus mucin treatment, and the lowest for minimal, but this is not significant. For biofilm formation trait, I did see a significant effect, but not in the direction that I expected. And so here I see the minimal and SCFM plus mucin have the highest variance within populations. And for twitch motility, again, no effect of treatment on variance within populations. But just to remind you, if you remember what I said earlier about what I observed in terms of the diversity of outcomes within a treatment, is that I saw more similar outcomes in the treatments without the spatial structure than with, and so I instead looked at the means within the populations of these phenotypic traits and asked, does the variance in the means depend on these treatments? And so here, so the blue dots are the means within those populations. And so, ignore the black line, which denotes the mean. What I want you to look at here is the variance in those points. And so, we can start to see that that the variance in the two treatments with mucin, the variance of the means is larger. And so this is for pyocyanin production, and for biofilm formation. Again, I saw the same difference in variance, much larger in the mucin treatments, and again for twitch motility, the same effect. And so the take-home message so far, there's still analysis to be done, but the take-home message for this talk is that I found no clear effect of treatment on diversification within populations, but I did find a pretty strong effect of treatment on diversification between populations. And I think this is a really interesting result that highlights the importance of contingency on diversification and adaptation. And so the next thing I plan to do is to sequence these populations to figure out the mechanism behind this and what the genomic basis of these differences in treatment effects on diversification are, which I'm very excited about. And so I'd like to thank you for your attention and thank these wonderful people for all the help they have given me. And I will take questions. I think I maybe have time for one.